Hello everyone, this is Dr. Hein Tun. I'm an assistant professor at the School of Public Health. Today I would like to bring all of you to my research lab. Let's go. So my research lab is working on two main projects in regard to the COVID-19 pandemic. So I would like to explain these two projects. The first project is we are trying to look at the gut microbiome of the people and how that is related to the immune response after the COVID-19 vaccination. So now I will introduce Dr. Tun, who is involved in this project, and can you explain a little bit about this project? Thanks.
Yeah, sure. So during our vaccine project, we recruited participants before and after they receive different kinds of vaccines, including Sinovac and BioNTech. And so we contact them to get their fecal samples. And so they will provide fecal samples under instructions through the videos we uploaded in the YouTube, and then put the fecal samples in that collection tube and sent to us for our microbiome analysis. And so this is used for their blood samples and for the immunoassays measurements. And yes.
So how did you profile the gut microbiome from the feces that you collected?
From the feces, so we will conduct a DNA extraction using the automatic DNA extraction machine, and then samples will be sent to hydroprobe sequencing. And so after analyzing the data, and we can try to associate the gut microbiome composition with different immunomodulation before and after the vaccine.
Great, so thanks. So this actually study has published the first paper in one of the top journal in the gut. So you can see here, so we also contributed the scientific knowledge to the community. So the second project that I would like to introduce is the surveillance of the SARS-CoV-2, which caused the COVID-19 pandemic, using the wastewater in the sewage in Hong Kong. So hi Daniel, so you are involved in this project. Can you introduce a little bit about the COVID-19 sewage surveillance project?
Definitely. So basically the wastewater project is split into three big stages. So the first stage is that the wastewater volume is pretty high. So what we first have to do is we have to concentrate down the viruses to extract it, and that relies on this ultracentrifuge machine right here. So this bad boy has been going on day and night during the pandemic to make sure that we get the right amount of virus load for us to extract. So that brings us to our second stage. So the second stage involves extracting the RNA from our concentrated virus samples. So that relies on an automated machine you might have seen over there on the way here. And last but not least, our third stage involves running qPCR to detect how much COVID RNA is inside the wastewater samples. And from there, we can then estimate around how much COVID there is in the samples, and then the authorities can make their judgment from there on out.
Great. So yeah, so this project actually is being very successful because this project contributed a lot to fight against COVID-19 in Hong Kong. So, and also the Hong Kong government supported quite a lot on this project, and even the chief executive also visited this lab. Yeah, very fascinating project. So actually, in addition to these two projects, we are also working on quite a lot of other projects in my lab. But hopefully, all of you have learned like these two projects briefly, and then hopefully see you guys soon in the campus. Thanks.